Hello students, good morning. Welcome back to a new video of Sunanda's tutorial. So today in this video we will discuss the cell division, cell cycle of cell division. So it is present in your plus 2 first year science biology syllabus under unit 3. So let's start. At first, what is cell division? Cell division means when a parent cell divides to form two or more cells, and that cells which are produced as a result of the division are called the daughter cells. So there are two distinct type of cell divisions, out of which the first one is called as the mitosis. Mitosis or equational division, in which chromosomes are equally distributed, resulting into two genetically identical daughter cells. So the first one is called the mitosis and the second one is called the meiosis or reductional division, which divides and form 4 haploid daughter cells. So if the parent cell have 16 number of chromosomes, after meiotic division or meiosis, 4 haploid cells are formed. That means 4 cells are formed with each 8 number of chromosomes. But in case of mitosis, if the parent cell have 16 number of chromosomes, at the end of the mitotic cycle, it will form two cells or two equal cells with the chromosomes which are identical to their parent cell. That means each having 16 numbers of chromosomes. So this is the difference between mitosis and meiosis. This is the basic difference. Mitosis generally occur in the somatic cells, and this meiosis or reductional division occur in general the reproductive cells during the gamete formation. Then what is cell cycle? Cell cycle refers to the series of events that takes place in a cell, resulting in the duplication of DNA and all the cell contents along with the cytoplasm. So these are an orderly sequence of events by which a growing cell duplicates its contents and divides into two. So what are the phases of cell cycle? There are two primary phases of cell cycle. One is interphase and the second one is M phase. Interphase is the most active phase of the cell cycle, in which the cell prepares itself for the cell division. The cells prepare itself for the upcoming division. And M phase is the phase where the actual cell division occur. So this is the diagrammatic representation of your cell cycle. So it is the interphase, which again divides into G1, S and G2 phase, and then the M phase or mitotic phase. Then the interphase is further comprises of 3 phases, which we have seen in this diagram. One is G1 phase, second one is S phase, and the third one is G2 phase. So what is G1 phase? It is the most metabolically active phase, and where the cell grows continuously during this phase. So what occur in the G1 phase? At first it is the most longest phase of the cell cycle, and during which the RNA or ribonucleic acid and proteins are formed. So and it is the first phase of your interphase. Then S phase. In S phase or synthetic phase, the DNA replication occur or synthesis of new DNA occur in this phase. And as a result, the DNA content per cell doubles. And in case of animal cell, in this phase, the centriole duplicates in the cytoplasm. It is the shortest phase of the interphase. Then in G2 or gap 2 or second growth phase, in which cell growth continues due to synthesis of RNAs and more proteins. So, but the DNA synthesis stops, and cell organelles like mitochondria and chloroplast also divides in this phase. So, the interphase is divided into these three phases. So then what is G0 or quiescent stage? What is quiescent stage? So in a specific point of G1 or growth 1 or gap 1 phase, a cell decides whether to start a new cycle or to withdraw from the cycle. This point is called as the G1 checkpoint. The cell that leaves the cell cycle to remain in a resting state or dormant stage is called as the G0 stage or quiescent phase, where the cell do not undergo further division and exits the G1 phase and enters into an inactive stage. This stage is known as the quiescent stage of the cell cycle. Then the M phase or actual cell division phase. This is the mitotic phase or phase of equational division, as the cell undergo complete reorganization to give birth a progeny cell that has same number of chromosomes as the parent cell. Other organelles are also divided equally by the process of cytokinesis, which is preceded by mitotic nuclear division. So the process of M phase again divided into two phases. One is karyokinesis and the second one is cytokinesis. Karyokinesis is the division of the nucleus and cytokinesis is the division of the cytoplasm. So at first in a cell, suppose it is a cell, nucleus divides to form two nucleus. Then the cytoplasm divides or separates in the process of cytokinesis to form two cells, two daughter cells. So the mitotic phase is divided into four stages. One is prophase, second metaphase, third anaphase, the last one is telophase. So the stages are prophase, metaphase, anaphase and telophase, PMAT. So these are the diagrammatic representation of different stages of mitosis. We will study these phases in details. So at first the prophase. So the nucleus of the cell becomes spherical and the cytoplasm becomes more viscous, that means more dense, more concentrated. Chromatin or the chromatin fibers which are present inside the nucleus slowly condenses into a well-defined chromosome. Each chromosome appears as two sister chromatids joined at the centromere. So these all events occur in the prophase. The spindle or microtubules begin to form outside the nucleus. In plants, the spindle apparatus or mitotic spindle is anastral. In animals, the mitotic spindle is amphiastral, which include two asters formed in opposite pole of the spindle. Each aster consists of two centrioles surrounded by astral rays. So what occur in prophase of the cell cycle. In prophase, the chromatin fibers which are appear like this, they condense to form chromosomes with two sister chromatids. Then outside the nucleus, the spindles begins to appear, spindle fibers begins to appear. In plant cell, they do not have the astral rays which are formed in the centriole, but in case of animal cell, they have centriole and they produce asters or astral rays in both the poles. So they are known as, the animal cells are known as the amphiastral. But in case of plant cells, they lack centriole, so astral rays do not form. The only spindle fibers formed in plant cell, and each spindle fibers are called as the microtubules, which consist of tubulin proteins. So what are the structures? During the late prophase stage, the chromatin fibers already starts condensing and appeared as chromosomes with two sister chromatids, and the spindle fibers starts appearing or begins to form outside the nucleus. In plant cells, they lack astral rays, but in case of animals, they have astral rays, and from that aster or centriole, the spindle fibers originates in case of animal cells. Then after prophase, metaphase is there. In metaphase, the nuclear envelope or the nuclear membrane has completely disintegrated. Two sister chromatids joined by centromere and positioned at the equator like this, referred to as the metaphase plate. All the chromosomes are arranged in a equational, suppose it is a cell, it is the nucleus, all the chromosomes joined and positioned in the center or equational plate or metaphasic plate. At the kinetochore, each chromatid is connected to spindle fiber from opposite pole. What is kinetochore? Kinetochore is a special protein or special protein complex that are present on the centromere. Suppose it is a chromosome and it is the centromere. A special type of protein is present associated with that centromere is called kinetochore. To that kinetochore, spindle fiber joined from opposite poles. Then after metaphase, anaphase is there, where the centromere splitting occur and two sister chromatids disconnect and travel to the opposite pole. Sister chromatids are now known as daughter chromosomes. So from the middle they split, and the separation of the sister chromatids occur, they disconnect from each other and travel to the opposite pole. Then telophase, chromosome starts decondensing when they reach the opposite pole. Each chromosome cluster develops a nuclear envelope around themselves and two daughter nuclei are created. Nucleolus, endoplasmic reticulum, Golgi apparatus all reappear. Then the cytokinesis. Cytokinesis, where the separation of cytoplasm occur after the formation of two nuclei in the process of karyokinesis. Cell organelles are shared by daughter cells. In some organisms such as fungi, algae and other plant cells, cytokinesis is not shortly followed by karyokinesis. A multinucleate stage is known as syncytium form, such as liquid endosperm of coconut, rhizopus, cenocytic hyphae and so on. where the cytokinesis do not occur. There only karyokinesis is present, and inside one cell many nucleus are present, and that multinucleate cell condition is called the syncytium form, which is found in the liquid endosperm of coconut, then cenocytic hyphae of rhizopus. So in higher plants and animals, there are two different methods of cytokinesis is present. One is cell plate method, one is cleavage or cell furrowing method. What is cell plate method? Cell plate method generally occur in plant cell, where the spindle fiber persists at the equatorial plane. The Golgi vesicles fuse at the center to form a barrel-shaped phragmoplast. Further addition of vesicles causes the phragmoplast to grow centrifugally till it meets the plasma membrane of the mother cell. The contents of phragmoplast solidifies to become cell plate or future middle lamella, which separates two daughter cells. The daughter protoplast secrete primary wall material on both sides of cell plate or middle lamella. So, suppose it is a cell. After the karyokinesis, two nucleus separates from each other. Now the cytokinesis occur. So in the process of cytokinesis or cell plate cytokinesis method, suppose it is a cell, these are two nucleus. At first, phragmoplast will develop in the middle and it will continue to grow in both the sides, and finally separates or attach, that plate attach with the cell membrane separating two cells, and finally two cells get separated in the process of cell plate cytokinesis. So here in this process, the phragmoplast grow centrifugally or apart from the center or towards the periphery. Then the cell cleavage method or cell furrow method, it occur in the animal cell or pollen mother cell of some angiosperms. In this process, a cleavage furrow appears at the middle, which gradually deepens and breaks the parent cell into two daughter cells. A special structure called mid-body is formed in the center, and it is a centripetal process. Suppose it is a cell, two nucleus is present. So in the middle, an ingrowth appeared and gradually it deepens, finally cutting two cells and formed this as two separate cells. So this is the cell cleavage cytokinesis or cell furrowing method. So this is the two types of cell cytokinesis method which appear in plant cell as well as in animal cell. In plant cell, cell plate method of cytokinesis occur, and in animal cell, cleavage cytokinesis or cell furrow method of cytokinesis occur. So it is all about your cell cycle, then mitosis process. In my next class, I will discuss the meiosis process, different stages of meiosis process. So please stay tuned, subscribe to my channel, Sunanda's tutorial, like all the videos and share with other friends. Thank you.